Hi everyone, I'm Liu. I'm currently a fifth year PhD of computational biology from Carnegie Mellon University. I'm going to talk about our recent work of a new tandem mass spectra data acquisition strategy called MS2Planner. Our lab is a dry lab for developing computational method of natural product discovery. The data that we usually use are metagenomics data and metabolomics data. One of the bottlenecks in our current pipeline is that the abundance of natural products are usually not high enough to be acquired by the current data acquisition strategy. So that is why we are very interested in developing new strategies that can improve the coverage of untargeted metabolomics. Currently, the most frequently used fragmentation spectra acquisition strategy for discovery is called data-dependent acquisition. Here is how it works. So this is a 2D space of mass to charge and retention time. The mass spectrometer will scan over the retention time axis, and for each slice of retention time, the DDA strategy will dynamically detect the most intense region of MZ. So here, the color region are the most abundant ones, and DDA will collect the precursor ions and get the fragmentation spectrum for each of them. And for the task of identification of small molecules, DDA can only collect the most abundant precursors and can only cover a small portions of the precursors. And even for those that are well above the detection limit of mass spectrometer, if their abundance is not higher than their competitor precursors, DDA will not detect them for further fragmentation. Another problem for DDA is that even if you repeat the DDA experiment on the same sample for multiple times, DDA still always collect the most abundant precursors and never improves the coverage. The goal of our new strategy, MS2Planner, is to not only collect the most abundant precursors, but also those low abundance precursors. In addition, we don't want to collect fragmentation spectra on the same precursor, but we want to maximize the number of acquisitions for distinct precursors. And if we want to run the experiment for multiple times, we also want to improve the coverage of the runs. And meanwhile, we don't want to collect the low quality fragmentation spectra, so we will also avoid the suboptimum intensity acquisitions. And in one sentence, based on a full preliminary MS1 run, we will schedule an optimum data collection path to maximize the number of distinct MS2 acquisitions while avoiding suboptimum intensity acquisitions. And one of the main difference between DDA and MS2Planner is that MS2Planner requires a full preliminary run of MS1. And based on the MS1 data, we use OpenMS to detect the apexes and cluster the raw MS1 signal according to these apexes. Assume we end up with n different clusters, and we can discretize the RT axis into T bins. Here, XTI is a binary indicator of whether we will acquire MS2 data at retention time T for cluster I. This corresponds to the last constraint of the optimization problem. Here, we assume that we will only schedule one data acquisition path, which means at each retention time, we will only collect the data for at most one cluster, and which is the second last constraint. This constraint can guarantee for each cluster, we will only collect data within a single retention time interval. In other words, we will not obtain the tandem mass spectrum of the same precursor for multiple times. The first constraint is designed to only collect high-quality fragmentation spectra. Here we use TIC, the total ion current of MS2, as a measure of the quality of the fragmentation spectra, and we will train a linear regression model to predict the expected TIC based on the sum of the MS1 intensity. And what we want to optimize is the total number of distinct MS2 acquisitions. In order to solve the optimization problem, we develop a computational method, MS2Planner. So here is a pipeline. We start from a full MS1 survey run and use OpenMS to detect the apexes and cluster the raw MS1 signal. And with this raw signal and clusters, we will construct a graph about the possible data collection paths, and we will discuss how to construct the graph later in details. Then we find the longest path of the graph so that the path can cover as many clusters as possible. In this way, we maximize the number of distinct MS2 acquisitions. And last, if we want to schedule multiple data collection paths, we can simply remove the nodes in the first path from the graph and then find the longest path in the remaining graph. This slide is about how to convert the optimum path schedule problem into a graph-based problem. In panel A, the black dots means raw MS1 signal, and the red dots means apexes. The raw MS1 signal are grouped according to these apexes. And after that, in panel B, in each cluster, some of the raw signals have the same retention time. So we further merge them into the same node, and the intensity of the nodes are the sum of the intensity of the raw signals. Panel C is about how to add edges to the graph within the cluster. For example, the cluster on the right. Remember, we have a regression model that can predict the total ion current of MS2 based on the sum of the MS1 intensity. So if the expected TIC of a retention time interval is above the threshold, and the expected TIC of any of its subinterval are below the threshold, we will connect the nodes with an edge. Here, between the nodes ut plus 4 and ut plus 6, the expected TIC is above the threshold, but between either t plus 4 and t plus 5, or t plus 5 to t plus 6, the expected TIC is below the threshold. Then we connect the node t plus 4 with t plus 6. Sometimes at a single retention time point, the expected TIC is always above the threshold. Then there's a self loop. For example, node t plus 7, which makes the graph not a directed acyclic graph. And in order to solve this problem, we split each node into an in node and out node. Here, U sub I indicates the in node, and U sub O indicates the out node. An edge should always go from an in node to an out node within the cluster. Between the cluster, the out node will point to the in node of the next cluster, as long as the retention time of the next cluster is greater than the current one. For the black edges within the cluster, we assign weight 0 to them. For the green edges between the clusters, we assign weight 1 to them. It can be proved that if we construct the graph in this way, the longest path of the graph is actually an optimum solution for the optimization problem that we mentioned before. Here are the 4 types of experiments we did for benchmarking. DDA-X means we use DDA mode with dynamic exclusion. MS2Planner DDA in X means we use DDA and targeted acquisition with inclusion lists generated by MS2Planner and dynamic exclusion. MS2Planner targeted in X means we only use the targeted inclusion list and dynamic exclusion. The last one means there's no dynamic exclusion in the targeted mode. So we compare these 4 strategies. For each of them, we run the experiment for 5 times on the same sample. The sample consists of NIH natural product reference molecules and some background fecal or tomato samples in order to increase the sample complexity. Here, the x-axis is the number of times a feature is identified, and the y-axis is the frequency of the identifications. And for the DDA and DDA with MS2Planner inclusion list, most of the features are identified for 5 times, and you can see the peak at 5, which makes sense because we run the experiment for 5 times. But for the targeted mode with MS2Planner inclusion list, more than 95% and 98% of the features are identified for just once or twice. This shows that MS2Planner can reduce duplicate acquisitions. Here in panel A, we show that over 5 iterations, targeted mode with MS2Planner inclusion list can identify more unique compounds than the DDA, indicating that MS2Planner can improve the coverage. In panel B, we use cosine similarity score to the reference spectra as a measure of quality of the spectra. When identifying the same number of compounds, the targeted mode with MS2Planner inclusion list, which are shown in red here, has higher cosine similarity score than the DDA modes, which are shown in blue. This means MS2Planner can acquire higher quality spectra than the DDA. We also compare the sensitivity and annotation rate of this method. The sensitivity means the number of ground truth molecules identified by the spectral library search over the total number of ground truth molecules in the sample. The ground truth molecules are the known NIH natural products molecules that we add into the sample. The annotation rate is the number of ground truth molecules identified by spectral library search over the number of unique MS2 collected by each method. And you can see that in terms of either sensitivity or annotation rate, the two targeted mode with MS2Planner inclusion list perform better than the DDA. To compare MS2Planner with DDA, in terms of spectral quality, sensitivity, annotation rate, coverage, MS2Planner is better than DDA. In summary, with alternate MS1 scans, DDA dynamically acquires the data, while with a full preliminary MS1 run, MS2Planner can schedule the globally optimum acquisition path. In addition, MS2Planner improves the coverage while maintains the quality of the MS2 acquisitions. MS2Planner also improves both sensitivity and annotation rate. I want to thank my advisor Hossein Mohimani and our collaborator Luis to lead the project. Zeyuan is a very efficient master student who implemented the code and wrote the draft of the manuscript. The funding of this paper is from Sloan Foundation, NIH, and Department of Energy. Thank you all for listening.